Hi guys, welcome to Lipids. So in terms of the triglycerides and phospholipids, you need to be able to explain the function and the structure. So we will be looking at the condensation and hydrolysis reactions. We will be looking at the different properties of triglycerides and phospholipids related to their function. And also you need to be able to describe the test for lipids, which is called emulsion test. So, firstly, what are the atoms involved in lipids? It's carbon, hydrogen, and oxygen. And really important information, lipids do not make polymers, okay? So they are not polymers. The ester bond that are found in the lipids, it's made between glycerol and each fatty acids. So what's the difference in terms of the atoms between lipids and carbohydrates? It's the fact that there is a smaller proportion of oxygen to carbon and hydrogen than in carbohydrates. They are insoluble in water, so that's important fact, but they are soluble in organic solvents like alcohols. And there are two main groups, triglycerides and phospholipids. So in terms of the function of them, they are the source of energy. So when oxidized, so what's the oxidation? Oxidation is a loss of electrons or hydrogen, but the gain of oxygen, and then the reduction is the gain of hydrogens or electrons, but the loss of oxygen. So when they are oxidized, lipids provide more than twice energy at the same mass of the carbohydrates, and they release valuable water. They are waterproofing because they are insoluble in water. They provide insulation because slow conductors of heat, and when they are stored penetrate the body surface, they help to retain body heat. So they act as electrical insulators. For example, you can see it in the myelin sheets around the nerve cells. So when you've got your myelin sheets, okay, the nerve impulse will be conducted in between those, so the distance, okay, that the nerve impulse need to take, it's much shorter rather than going through the whole length of the axon. They also provide protection stored around the organs, for example around the kidney. So in terms of the structure, there are the triglycerides, so easy to remember, okay, refers to number 3. They are made of 3 fatty acids, so those are fatty acids. As you can see, it's a chain of carbons and hydrogens combined with the glycerol, which is here. Here we've got the ester bond. So ester bond, it's made between each glycerol and fatty acids. How is it made? It's made by the condensation reaction, so the removal of water. And if we were looking then at the hydrolysis of the glycerol, so the glycerol will be hydrolyzed, okay, to 3 fatty acids. The triglycerides will be hydrolyzed to 3 fatty acids, to glycerol, and obviously, and that's the structure. So in terms of the R group, so other words, in terms of the fatty acids, which we call them R groups as well, they could be saturated or unsaturated. So saturated, it's this chain here, for example. So what's the important thing? There is no double bonds between carbon atoms. So it's fully saturated with the hydrogens, has the highest amount of the hydrogens. Unsaturated fatty acids, then they have at least one double bond between the carbons, hence they have less hydrogens. We can have monounsaturated fatty acids, which contains only one bond, which is present here, or we can have polyunsaturated fatty acids, which will contain more than one double bond between carbon atoms. In terms of the relationship of the structure to function, again our so what man coming along. So remember, you need to use the so what approach. There are 4 main aspects here. So they have high ratio of energy storing carbon-hydrogen bonds, which we've seen, and this is to make it an excellent source of energy. They have low mass to energy ratio, so a good storage molecule because much energy can be stored in small volume. They are large, non-polar, insoluble in water, so the storage does not affect osmosis, does not affect water potential. Okay, and the high ratio of the hydrogen to oxygen atom, which we mentioned, so release water when oxidized, and provide an important source of water. So phospholipids then, so this is second type of the lipids. There are two fatty acids in here combined with glycerol and the phosphate molecule. So the main difference is the fact that one of the fatty acids is replaced by the phosphate in phospholipids, but in triglycerides you had three fatty acids and the glycerol. So fatty acids will repel water because of the hydrophobic tail. Okay, so in terms of the tail, we are referring to fatty acids, and they will be oriented itself away from the water. Okay, so it will be like in the middle here, and then we've got the phosphate molecule, so that hydrophobic head that can interact with water but not with fat. So, and there are polar molecules. So now the relationship again to the structure with so what approach. So there are polar molecules. So in the watery environment, phospholipids molecules will form a bilayer between the cell surface membrane. So the cell surface membrane of each cell is made of the phospholipids in the manner that the phosphate is on the outside and then the fatty acids are inside. So the hydrophobic phosphates of the head help to hold the surface of the cell surface membrane, and they also can form glycolipids by combining with the carbohydrates within the cell surface membrane. And when you will be learning about immunity, glycolipids are really important in cell recognition. So, question, describe the difference between the structure. So again, difference. So you need to talk about two things, what they want. They want triglycerides and they want phospholipids in here. And what's the focus? It's the structure, only 1 mark. So what's the main difference? It's the fact that in phospholipids, one fatty acid is replaced by a phosphate, but in triglycerides, we've got 3 fatty acids. Here we've got a question, okay? So we've got the phospholipid. How can we recognize this? Because we've got the phosphate. So the part of phospholipid labeled 8, which is here, okay? What is it? It's a glycerol. Name the type of bond between A and X. So remember, between each glycerol and the fatty acid, we have the ester bond, and which of the fatty acids, X or Y, is unsaturated. So unsaturated means has a double bond. So double bond we've got in Y. So has a double bond between carbon atoms, but you need to say where the double bond is. So don't be silly. Remember, say between the carbon atoms in that chain. So emulsion test is then the test that you're going to use for lipids. How to do it? So you're adding your sample. You're going to add ethanol because remember, lipids are insoluble in water but soluble in ethanol. You're going to shake it, and then you're going to add the water, shake it gently. So you're going to get as a result, positive result, the cloudy white color. So that will indicate the presence of lipids. Why is this coming from here? Because obviously the lipids are soluble in ethanol but insoluble in water. So oil droplets, okay, from oil droplets to water droplets will make this appear cloudy. So that's the test. And the last question here, we've got to describe